So next I want to talk about the bacterial transformation. Previously I covered what plasmids are and how you use plasmids to transform bacterial cells. And pretty much what you do is you just lyse the cells, so break down the cell membranes in order to extract the plasmids from different bacteria. These can be found in nature or you can make them yourself. And then afterwards you're going to somehow, I'll get into it, you'll put the plasmid in another bacteria and they'll uptake it and start emitting that gene by transcription and translation.
So the plasmid contains DNA.
Yeah, it's just a circular DNA strand that gets transmitted between bacteria. It's how they, give genes to each other. So predominantly what we do in lab in order to replicate plasmids and use them for other things is through a thing called transformation, and we mini prep them. So I wanted to talk more about transformation and that protocol that we use in lab in order to do it. So transformation, at least in iGEM, is split into two main things: chemical transformation and electrical transformation. I've kind of gone into this before, but in chemical transformation, you're going to use heat shock to open up that cell membrane in order to let the plasmids through, so the bacteria can adopt it. But it's less efficient in terms of transformation efficiency. So not all the bacteria that you throw or not all of the plasmids that you throw into that bacteria and are going to be properly transformed. So some of the bacteria are going to have the plasmid, some of them are not. And in order to determine which one doesn't and which one does, you're going to do a process called screening, which you do on plates.
And heat shock doesn't damage the DNA?
Well, it would if you increased it like incredibly. So we're doing heat shock at around 55 C, which I'm sure if you left the DNA in there for an extended period of time, it would denature. But we only leave it in there for 45 seconds, which is not enough to kill the cells off or destroy the DNA. I'm sure some does, but it's not like crazy inefficient. But chemical transformation is really easy compared to electrical transformation, and it's cost efficient. So with electrical transformation, you're going to need to use electrical current to open up those pores that within the cell membrane to let those plasmids go in. It's more efficient, but it costs more because you have to have a device called an electroporator, which we technically have. I do not have any experience using it though. We do have electrical competent cells that we can use. So if any of you have interest using those cells or learning how to use the electroporator, I'm all up for it. Okay, because it's much more efficient in terms of transformation efficiency. So I'm just going to go into chemical competence and transforming cells via the chemical means because that's what we predominantly use in lab. So first, you're going to want to choose your plasmid of interest, your gene of interest, in order to insert that into the bacteria. That can be found in the iGEM registry. We have a bunch of kit plates that I went into last week where you can pull these plasmids out in order to resuspend them and finally put them back into the cells. Next, you're going to gather the chemically competent cells from the negative 80-degree freezer. That freezer is currently in, I think, G35. You guys shouldn't have access to that, but I do. So when we work, I'll maybe even take you to that lab. I can show you around. We can open up the freezer, take out what we need, and leave. Typically, I do not work in the G35 lab, though, because it's a tissue lab, and there's a bunch of professors and grad students working in there. I don't want to mess with them with the stuff I'm doing. Afterwards, you're going to want to start combining the DNA with the cells. You're going to take a specific amount of cells out, a specific amount of DNA, combine them together in an epitube, and you're going to want to incubate that for around 5 minutes in order to get that nice and mixed together. Next, you're going to want to heat shock them. So this heat shocking is what opens up the cell membrane, and that's going to allow the DNA to enter. Next, you're going to want to incubate them again, except this incubation is on ice. So I specified with the asterisk when incubation's on ice and when it's just in 55 degrees Celsius water. So typically, you're going to want to keep the cells as cold as possible because if you leave them out in a warm environment, they're going to start to degrade. So the only time you're going to incubate them in a hot environment is when you're either growing the cells on a plate or you're heat shocking them. Every other time, you're going to want to have these cells on ice as much as possible. And when you're holding them in their hand, try not to touch it, like touch the cells themselves with your hand. Hold it from the top of the tube, because otherwise your body heat's going to start degrading the cells. After they're incubated for another 2 to 5 minutes on this ice, after you did the heat shock, you're going to want to throw SOC broth, so this stuff right here. SOC broth is a recovery medium. It comes with the intact genomic cells that we order. It's pretty much just a really nutrient-dense broth that gets the cells back up to functioning. So you can actually start plating them on antibiotic because the antibiotic would kill them otherwise. So you want these cells to be as strong as possible. Finally, you're going to want to incubate these for an extended period of time. So that's around an hour to an hour 30 just to get that SOC broth mixed nice and good. And you're going to want to spread those cells on Petri dishes in order to start screening for them. That's typically for around 8 to 12 hours, though I typically don't have that much time, or I don't have that sequence of time, so I have to come in like the day after, the morning after, to pick these things up and start screening for them. So these are the E. coli cells that we have. I think it's a different strain of E. coli, but they look exactly the same. You can see the bacteria at the top. Pretty much you just wait for it to thaw out, you extract it and put it in another container so you don't break the other cells. When you're pipetting it out, make sure to be as careful as possible. If you pipette too quickly or too hard and you're like going up and down, that will break the cell membranes and your cells will be useless. So now, I apologize for this. I really wanted to go into the protocol just to introduce you guys to it. It's kind of death by PowerPoint though. I try to space things out as much as possible. It's a 10 to 11 step process. Compared to other protocols that we have in lab transformations, not all that complicated. You pretty much just use pipetting, you mix some stuff together and you throw it, incubate it. It's not that bad. So first you're going to want to gather 1.5 milliliter epi. This is going to be your predominant storage unit within the transformation protocol. You're going to want to grab your chemically competent cells on ice and also your plasmid DNA of interest on ice. I also recommend putting the epi, the empty epi, on ice too, because that gets it up to temp, so it's not room temp when you put the cells in. So just put everything on ice. And in order to get these cells, you'll have to ask me for the negative 80-degree freezer. However, the plasmid DNA is in our G6 lab. It's in the freezer in the back. So typically you're going to want to pull that out last, because otherwise it's going to be in the ice for a very long time because you have to go to the other lab to pick up the cells. Next, you're going to want to pipette out 50 microliters of cells into the 1.5 milliliter epi tube. I already said this, be careful because you could break the cells. You're going to want to add 1 to 2 microliters of DNA, plasmid DNA, to these cells. You can mix by tapping it a few times. Just do not vortex it or shake it around because once again, they're fragile. Most of the time we'll be using kit plate DNA. Do you know what that is?
Kit plate? No. Is that like agar plates?
So last week I went over kit plates. Kit plates are pretty much just our fundamental storage unit for all different types of plasmids. Okay. The iGEM organization sends out these kit plates every year, and it's just hundreds of parts that you can use. And all these parts are documented on their website. So you go to their website, Google what part you want. It tells you where it's located in the kit plate itself, and you can just take it out and transform some bacteria with it. Okay. So typically we'd be working with kit plate DNA when we start fitting this DNA into the cells. But if you do a successful ligation, so start gluing bacteria together, gluing DNA together, and you want to transform cells with your newly created DNA, you're also going to use this procedure for doing that. Mix by tapping, incubate on ice for 30 minutes in order to get that. I said 5 minutes earlier. It's actually 30 minutes. That's in order to get it nice and prepared for the heat shock, so they're not like completely, broken apart when you heat shock them.
Okay.
Okay, step 5. You're going to heat shock the cells at 42 degrees Celsius, not 55. 55 is for, what is it for? Oh, it's for gel electrophoresis when you're incubating with gels. So 42 degrees Celsius. Right now we don't have a water bath because it went missing, so we'll have to go to another lab in order to do the water bath stuff.
It went missing? Yeah,
grad students share our lab, and sometimes the grad students don't know that the equipment's technically ours, and so they walk out with it, take it to their other lab they're working with. Because technically our lab is a storage room along with being our lab. We're working on it. Right now, I'm working with Dr. Finnegan and getting a new water bath for us.
Okay.
When you're doing this, I recommend using a stopwatch or a phone timer just so you make sure it doesn't go over a minute. After a minute of being in 42 degrees Celsius water, that will start to denature, so you want to pull it out as quick as possible. And immediately, once you pull it out, put it back on ice for another 2 minutes in order to solidify the cell membranes a little bit. After you wait those 2 minutes, you're going to want to add 950 microliters of SOC broth. Right now we have, I believe, 3 or 4 containers of SOC broth within the MST iGEM fridge. It's about, I would say, 2 to 3 milliliters per container. They'll last forever because you're only using about a milliliter. So pipette that in there. You can mix again by tapping, just don't be super rough with it. And what this SOC broth is used for is to strengthen those colonies for plating afterwards. Finally, if we have a shaking incubator open, you're going to put that SOC broth cell mixture in the shaking incubator at 37 degrees Celsius in order to get those start to grow and start to exponentially grow. Those incubators are stationed in the microbiome lab, shrink 128. We technically do have a shaking incubator in G6. However, it is broken, and the only reason why it heats up at all is because the motor creates heat when it runs. That gets around 33 to 35 degrees Celsius, which technically works. It's just going to decrease the efficiency a little bit. I recommend using the incubators in 128 though. Finally, this is pretty much the last step. You're going to want to spread the bacteria on plates, put them in an incubator, and just grow them overnight in order to see if you have any colonies that were properly transformed or not. And in order to screen them, you're going to want to have antibiotic on those plates. And so what the antibiotic does is, if you have antibiotic on it, and if the cell has the DNA, that cell's going to have that antibiotic resistance. So the cells that have the antibiotic resistance will grow on these plates, while the ones that don't won't. And so you can start extracting the colonies that grow on it, and then you can take those colonies, throw it in a broth culture, make tons of colonies, and then extract that to make even more DNA. So it's like a self-perpetuating loop where you get more and more DNA, so you never run out of stock. Right now, we've been working a lot with the mini prep protocol. That's what it's called. It's called mini prepping DNA. And I haven't had really much success with it myself with the parts we want. However, RFP, red fluorescence protein, it works like every time.
Seriously?
Yeah. So for some reason, the parts I'm working with, red fluorescence protein, just works every time. I have colonies that are red growing on it, and all the other parts don't seem to grow at all. So right now, we're kind of in a mixture of doing restriction digests, so getting these parts cut properly, and also getting mini prep so we don't run out of parts.
Okay.
So I think it's tomorrow, I'm going to be working with Nick, and we'll be doing transformations in order to try to get into the beginning stages of doing mini preps. And you said you were open Saturday, so I guess if it works, even if it's just RFP that works as our control, we'll be inoculating just to get you experience and myself more experience.
Okay, cool.
Yeah.
What do you have like a specific time on Saturday that I can be there?
I typically work like it from 1 to 2.
1 to 2? Okay.
Well, starting from 1 to 2.
Yeah, and then we're gonna be down in G6.
G6, yeah.
Cool.
The procedure itself can take anywhere between, I would say, an hour and a half to 3 hours.
Okay.
So there are 2 different types of transformation protocols. There's the 5-minute protocol, and then there's the 90-minute protocol. The 5-minute protocol has a shorter or a much smaller transformation efficiency. It's like minus 10% or something like that. While the longer one, of course, has higher transformation efficiency. When I don't have very much time, I just run the 5-minute, which is actually more like 45 minutes to an hour. And when I have enough time, like say Saturday, we'll be running the 90-minute protocol, which is probably going to take around 3 hours.
Okay.
So the rule of thumb there is, if you have a procedure and it says a time limit, multiply that by 2, and that's most likely how long it's going to take. I just wanted to get into a debrief on mini prepping and what screening is. Next week, I'll go into that in a lot more detail, because it's a very complicated procedure compared to transforming cells, and there's a lot of room for error when you do it. But I just wanted to introduce it to you to get you prepared for next week. So screening is the process of growing colonies on an antibiotic medium, which is, in our case, is going to be LB agar, lysogeny broth, with agarose, which is just Jello. It's the standard, it's in Petri dishes, and you just spread the bacteria on it, and you grow it overnight. So when the bacteria are not properly transformed, they're going to die on the plates because they do not have the gene. And when they are properly transformed, they will grow on the plates because it has the antibiotic gene. And each independent successful colony that grows on the plate all have the same DNA because it came from one cell. And so when these cells grew, they exponentially grew, divided again and again and again through mitosis. And finally, you have a big colony. It's typically round unless you have crazy growth on the plates. And you can extract this colony, and you know in that colony, every single cell has the same DNA, which is helpful because you don't want to be mixing different E. coli with different DNAs in the same culture when you're inoculating them. So the easiest way to harvest transformed cells in order to not run out of parts is to pull from one growing colony and only that colony. Don't be pulling from multiple colonies and throwing it in a broth medium. So these are some of the RFP plates I grew. Notice I wanted, what are some failures with this? Because there's a lot of things wrong with this image.
Well, let me see. I can see that in a lot of, the circles, that's where the antibiotics are put into the plate, right?
So this is a colony. This is an RFP
No,
colony.
I mean the circles in the clear. You see them?
Oh, these?
Yeah, that's like the antibiotics, right?
No, this is just the.
Oh,
This
sorry.
is just the bottom of the incubator. Yeah.
So you're asking like what I see?
Yeah. What's wrong with these colonies?
I mean, they're growing pretty spottily.
Yeah. Well, that's one thing. I didn't have much growth on these, quite surprisingly. That means my transformation efficiency was pretty low. Typically, when you spread these, you're gonna have like some colonies over here, maybe some over there.
So I don't know how you straight, but the way I was taught to straight is you go down and you
Yeah.
rub it there and then you rub it there. It seems to only be growing in the part that like is supposed to have like the most bacteria. It's not growing in the parts with less.
Yeah, exactly.
Yeah.
So you can see right here, I kind of did the streaking a little bit.
Yeah.
In the past, I used beads, and that's typically what we use, but currently we're out of beads to spread. I think they're all dirty.
Okay.
They need to be autoclaved. Another thing is they're all clustered.
Yeah.
So this is what we call, it's very similar to what we call lawn growth, where you just have pretty much an entire field of bacteria, and it's really hard to separate them in order to only get one type of cell that has a certain type of DNA. So when stuff like this happens, it's really bad because you don't know if what you're pulling from has multiple different types of DNA or if it's just one.
You want it to be
Because especially like this colony, I can't tell if that's one colony or 10.
Yeah.
Typically, the reason why these are like spaced out like that is because they have different DNA, that the E. coli are going to want to fight each other. They're going to want to try to outcompete one another. So that's why they're spaced out in multiple colonies. But for some reason, these got super, super close, which is bad, and that's lawn growth. So typically when we're in lab, we don't want stuff like this. We want stuff more like this, but spread over the entire plate. This could be, the source of this could be many things. I personally think it's the cells themselves. The transformation efficiency of the cells are pretty low. They are from Intact Genomics, which is a St. Louis-based company, and they say it's good. We've worked with them in the past, but when I talked to Dr. Westenberg, one of our advisors, he's like, when he looked at this, he's like, this screams transformation efficiency cells. They're really low. That's what this screams to me.
I mean, did you talk to the company?
No, I did not.
I mean, you could have. Maybe there's something wrong with the lot.
Yeah, it also could have been they were shipped here and they were shipped in dry ice, and they could have been, transit problems. Other than that, at least they had growth. So these plates, even if they have spotty growth like this, you can still use them because you can pull from colonies over here. My other plates that had the parts that we're using for our project, so we need a promoter for our project that I was trying to grow. Those did not grow on the plates at all. There was not a single colony, which really confuses me because they're in plasmids with the antibiotic resistance that we need. Why are they not growing on these plates? Dr. Westenberg thinks that it's just transformation efficiency that is super low, but that doesn't make sense to me because the DNA that I used was of extremely low concentration. So typically you want to work with around 100 to 200 nanograms of DNA per 50 microliters of cells. And this RFP, it was like 3.
Yeah.
It was like 3. And these still grew and they still properly transformed. So that tells me that it's not a problem really with transformation efficiency. There's something wrong with the parts themselves. Like either they're mislabeled, they don't have the right antibiotic resistance, or the part is somehow conflicting with the cellular operations of the bacteria. It's somehow inhibiting them, preventing growth. Now, if you guys have any ideas on that, or if you want to look into it.
I would just talk to the company. I know where I worked, if there was something wrong with the lot, we kept extra samples and we had testing available. We were able to test to see if there was something wrong with the lot.
Yeah. Okay.
So, I mean, you could contact the company.
Yeah, I don't know. it's a whole debugging procedure. I have a lot more ideas with debugging gel electrophoresis than this itself. This is one of the things that our iGEM team in the past has struggled with the most. One of two things: gel electrophoresis is not bad in itself, but restriction digests, what you do before gel electrophoresis, cutting the DNA.
Yeah.
We've always struggled with that, and we've always struggled with mini preps. But typically for new teams, mini preps is where they always struggle at, because you don't know. It's a pretty simple procedure in doing this, and because it's so simple, you have no idea what's going wrong.
I mean, there's also a lot of steps, and there's also a lot of places where things could go wrong. That cells could get denatured at many different places.
Yeah. I've done the procedure so many times that I believe I'm doing the procedure right. So I think it's the materials or the DNA itself. Anyways, next slide. So next week, I'm going to discuss mini prepping in more detail. That's, once again, extracting DNA from the cells in order to use it for other things like digests, like ligations, stuff like that. And I just said this, mini prepping is one of the things we struggle with the most here.